Hello, good morning to all. Uh, Today we are going to discuss about the topics including pseudogenes, gene families, gene clusters, and gene superfamilies, which is coming under the molecular biology syllabus of MSc Microbiology of University of Calicut. Now, in the case of a genome, especially a eukaryotic genome, you know that there are various regions in the genetic material which may be coding, non-coding, junk DNA, or some other regions of the DNA which are referred to as non-coding regions, which contributes to majority of the total genome size. So the genetic content of an organism, in short, we call it as genome of an organism, that includes the genes. The genes may be coding or non-coding regions like introns and exons, which is present in the gene, which contributes about 30% of the overall size of the genome. And the gene which contains the coding regions or the exon regions is only about 1.5% of the total size of the genome. In case of humans, the overall coding regions, which corresponds to about 25,000 to 30,000 genes, which is identified, contributes to only about 2% or less than 2% of the size of the genome. So this is the general tendency. Whereas the other elements like transposomes, which is also known as jumping genes or transposable elements, which is reported to move from one part of the genome to another part of the genome by itself, they contribute about 45% or a major share of the overall size of the genome. Then there are other miscellaneous elements like SSRs or STs, and various other elements were also present in the genomic content. The lines, which is also known as long interspaced elements, these are a group of genetic elements that are found in large numbers in eukaryotic genomes, comprising about 21% of the human genome. And 99.9% of which is no longer capable of any retrotransposition and therefore considered as dead or inactive. So basically they are coming under the transposable elements, but the lines were not having anymore the ability to jump from one position to other. That is, they lost the ability to do the retrotransposition activity. So they are considered as dead or inactive. And these lines contribute about 21% of the eukaryotic genome. And we can say that 17% to 21% of the human genome is reported to have these particular elements of long interspersed elements or lines. And 13% is contributed by short interspersed nuclear elements. And these are sequences of non-coding DNA present in high frequencies in various eukaryotic genomes. And they are a class of retrotransposomes. And these retrotransposomes, these DNA elements that amplify themselves throughout the eukaryotic genomes, are often through RNA intermediates. Signs or short interspaced nuclear elements are sequences of non-coding DNA present in high frequencies in various eukaryotic genomes. And they are a class of retrotransposomes, the DNA elements that amplify themselves throughout the eukaryotic genomes, often through RNA intermediates. The SSR markers, which is coming under the miscellaneous category, microsatellites, otherwise called simple sequence repeats, or short tandem repeats, which is also known as STRs, are sequences of 2 to 5 base pair length of DNA, and it is a sort of variable number tandem repeat VNTR, and these SSR markers are important in various genetic studies. The satellite DNA, which is also marked as SD in the diagram, these are short sequences which are not capable of any transposition, and especially it is found in the centromeric regions, often found in centromeric regions, and they are repetitive nucleotides which is present in the centromeric regions, and they don't have the ability to perform the transposomes. So these are all different categories of the DNA or genetic content that contributes to a major share of a eukaryotic genome. Now when we come to the structure of a human chromosome, take the example of chromosome 22, which is having about a size of about 48 into 10 raised to 6 nucleotide pairs of DNA, which is present in the chromosome 22. About 10% of the chromosome is supposed to contain about 40 genes. The chromosome 20 of the humans, the overall size of that, if you are considering that size, 10% of the chromosome 20 is supposed to contain about 40 genes. Again, we can, if you are taking 10% out of that, 1% of the chromosome may contain 4 genes. So this 1% of the chromosome 20, which is supposed to contain about 4 genes, will have its own regulatory sequences, introns, exons, and various other regions or intron regions, which is supposed to be removed, and junk DNA, all these things will be present in between that 1% along with the 4 genes that is present in that 1%. So these genes, the functional part, the exons, will be joined together when you are transcribing something. That will be transcribed, and that introns will be removed, exons will be joined together, then the concerned protein will be translated and it will be folded, and that will be expressed. So this is the structure of a gene and the overall DNA content which is present in the chromosome. So in chromosome 22, if you are taking as an example, the quantity of the DNA or the number of genes which is present in that particular chromosome in 1% is about 4 genes. That is the approximation. Okay. So as we discussed, the genome, eukaryotic genome, contains various types of genetic elements, including genes, coding and non-coding regions, then transposomes, lines, signs, etc., which is coming under the transposomes, and of course the miscellaneous elements, including the VNTRs and satellite DNA and all other things, all is a part of the overall genome components. Now when we discuss different categories of the DNA that is present, as the title indicates, we have some components like pseudogenes, gene families. We have to identify what is that, what does it mean. In case of pseudogenes are a family of genes which is derived from a common ancestor due to a gene duplication event. Pseudogenes, as I mentioned, it is a family of genes. Family means it is having a common ancestor. So these genes actually they they have a duplication, gene duplication event normally. Here we can see that the typical gene is presented with regulatory elements or control region, two exon components and intron or non-coding region which is in between. So this is a typical gene which is present in a eukaryotic system. From this gene, the RNA is transcribed. RNA is processed after the post-transcriptional modification by removing the introns. Then it is this RNA, processed RNA is translated into the corresponding protein and that particular function is expressed. This is how a eukaryotic gene works. Now, in the case of a formation of a classical pseudogene, this gene may be duplicated and now we have two genes, a duplicated gene present in the immediate vicinity of the first gene. So these two duplicated genes may undergo or one among them may undergo a mutation in one copy of the gene which deletes the regulatory element or remove the regulatory element. The result is that one will be a functional gene, the other one will become a pseudogene because it doesn't have the ability to be coded, but the sequence is there, but it will not be functional or it will not be coded because the regulatory elements were lost during the mutation event. So this becomes a pseudogene because the sequence is present, but it cannot be expressed. Whereas the other one, the functional copy may be there and it will be useful for performing the normal functions of that particular gene, but the other copy that is actually mutated and lost the ability to express due to the lack of these regulatory elements will become a pseudogene. So this is one classical example or classical mechanism of formation of a pseudogene, and there may be other mechanism. And the second mechanism is reported to be the formation of pseudogenes from processed genes. Processed gene means here in the case of the normal gene, the RNA is expressed, it is processed and the intron or intervening regions were removed in the case of the processed RNA. And a reverse transcription event of this processed RNA will result in production of a copy of a RNA or a DNA copy of the corresponding processed gene. And subsequently, this may be inserted back into the genome. So this event of insertion of a processed copy of the target gene back into the genome by the process of reverse transcription may create a pseudogene. Here you don't have the regulatory elements, so it cannot be functional, but the sequence is inserted in between the chromosomal DNA. So that becomes a processed pseudogene. So this is a normal pseudogene, classical mechanism of production, and this is a mechanism of production of a processed pseudogene which is resulting from the reverse transcription and subsequent integration into the genome of a processed RNA. Now see this slide. There is a functional gene, and this functional gene may produce a functional protein with a normal amino acid sequence. Or from this functional gene, an insertion or deletion mutation, indel mutation will produce a pseudogene. Pseudogene may not produce a functional protein. So the pseudogene, which is a dysfunctional gene produced by an indel mutation from the functional gene, will produce a nonsense protein because an insertion or deletion will result in the frame shift mutation, and that frame shift will change the entire sequence of the amino acids that is being coded from that particular sequence. So it will be a nonsense protein or non-functional protein which is coded if it is doing something like that. Now, here we have another option where, so this is an indel mutation, and in the second case we have the functional gene, and that functional gene, it will be subjected to a nonsense mutation, and from that nonsense mutation a pseudogene is produced. The nonsense mutation will insert a termination codon. Instead of shifting the frame, it will result in the production of a nonsense codon, and subsequently that particular protein is the production or the translation of the particular protein is interrupted at this point. So this also result in a pseudogene which prevents the functional protein to be produced from that gene. So we have indel mutations which produces a nonsense protein or an unwanted protein, or a nonsense mutation which results in or which prevents the protein from being coded by inserting a nonsense mutation in the actual gene. Why these pseudogenes? They're simply a historical accident, and they represent another category of junk DNA. Perhaps not. There is growing evidence that pseudogenes can be transcribed into RNA, and this RNA can be or cannot be translated into protein, but can provide sequences into which microRNAs or mRNA can bind. The microRNAs, they bind to the mRNA, inhibiting their translation and accelerating their degradation. In either case, they suppresses the gene expression, that is the synthesis of the protein. But pseudogenes, they are competing with the same miRNAs as their functional parent. So the repression of the functional gene is relieved because the miRNAs and pseudogenes will be competing each other, and subsequently the parent RNA, microRNA, will be relieved from the repression by the miRNA. So these miRNAs will be removed by the competing pseudogenes. Usually, if there is only one copy of the functional gene which is suppressed by or blocked by that particular miRNA, then there will be silencing of the gene or suppression of the gene expression. But if you have multiple copies along of the pseudogenes along with the functional gene, then the miRNAs has to block the functional gene and also the pseudogenes. So the available miRNAs will have to compete with the pseudogenes to reach the functional gene. So the result is that the functional gene will be relieved of its repression by the miRNAs. Okay. So if the functional gene is a proto-oncogene, its elevated expression can lead to cancer. So such a pseudogene represents another category of oncogene. There is evidence in both mice and humans of cancers that are driven by the elevated expression of pseudogenes related to a non-proto-oncogene. These are the properties of the pseudogenes. We can summarize it as they are ubiquitous and abundant in genomes. Then they are once called genomic fossils and treated as junk DNA. Then they play an essential role in gene regulation of their parent gene, and many pseudogenes are transcribed into the RNA. Then pseudogene transcripts may also form small interfering RNAs or decrease cellular miRNA concentration. They regulate tumor suppressors and oncogenes. So these are some of the properties of the pseudogenes. Now when we come to the gene families, what are gene families? A gene family is a group of genes that share important characteristics. In many cases, genes in a family share a similar sequence of DNA building blocks or nucleotides. These genes provide instructions for making products such as proteins that have a similar structural function. In other cases, dissimilar genes are grouped together in a family because proteins produced from these genes work together as a unit or participate in the same process. So this is an example of a gene family. Here we have the globin gene family. There is an ancestral globin gene, alpha and beta, and subsequently they branched and formed the alpha and beta globin gene family. So this is resulting from a gene duplication event of the ancestral globin gene. Then subsequent mutations resulted in the production of multiple or different types of or variants of that particular family members under the globin, alpha globin and beta globin family. So these family members actually they share a common ancestry, evolutionary ancestry is there for all the family members of the gene family. So here we have another example in the case of these chromosome 16. Here we have alpha gene, alpha 1, and this is a sequence and the different regions of the introns and exons and enhancer of the alpha gene family. And here in the chromosome 11 we have the beta, and these are the different regions including the promoter region, intron and exon and enhancer regions of the beta gene family. And this is having a common structure, but from the resultant mutations accumulated during the evolutionary time period, they have different functions and even different structure, but still they are classified in the same family, gene family. Now, classifying individual genes into families helps researchers to describe how genes are related to each other. And researchers can use gene families to predict the function of newly identified genes based on their similarities to other non-genes. Similarities among genes in a family can also be used to predict where and when a specific gene is active or it is expressed. Additionally, gene families may provide clues for identifying genes that are involved in particular disease. Sometimes not enough is known about a gene to assign it to an established family. The current understanding on that particular gene may not be sufficient for placing it under a family. In other cases, genes may fit into more than one family. It will have different family members. And no formal guidelines define the criteria of grouping these genes together. The classification systems of genes continue to evolve as scientists learn more about the structure and function of genes and the relationship between them. Now, a gene cluster. A gene cluster is a part of a gene family, but a gene cluster is a group of two or more genes found within an organism's DNA that encode for a similar polypeptide or protein, which collectively share a generalized function and are often located within a few thousand base pairs of each other. And the size of the gene clusters can vary significantly from a few genes to several hundred genes. So this is about gene cluster. So a gene cluster, it is a group of two or more genes found within an organism's DNA that encode for similar polypeptide. So two or more genes should be there within the same organism that encode similar polypeptides or proteins which collectively share generalized function. There should be similarity in the structure of the peptide, similarity in the function, and it should be present in the same organism. So these type of genes are known as gene clusters, and they are located within in the very close proximity also. Then only you can call it as a cluster. If one gene is present in chromosome 21 and another one is present in chromosome 3, then that is not a gene cluster. Gene cluster, or the members of the gene cluster, which will have the similar structure, similar function, and placed together or in the very close proximity of the chromosome, is known as a gene cluster. And the size of the gene clusters can vary significantly from a few genes to hundreds of genes, which is placed as a cluster. Now, a portion of the DNA sequence of each gene within a gene clusters are found to be identical. Not the complete sequence, but at least a portion of the DNA sequence of the members of the cluster will be identical. However, the resulting protein of each gene is distinctive from the resulting protein of another gene within the cluster. So they are identical or they are similar, which will have some common domain or common or similarity between the different members will be there, but it is not the same. Same protein is not there, but functionally and structurally and functionally it will be related. The gene found in a gene cluster may be observed near to one another, or on the same chromosome or on different but homologous chromosomes. An example of a gene cluster is a Hox gene, which is made up of 8 genes and is part of a Hox gene family. So one example is a Hox gene family where we have 8 genes which is placed as a cluster. So this is the example of the 8 genes which is placed as a cluster in Hox gene family. Multi-gene families and superfamilies were another group of genes which is of importance. The multi-gene families are defined as a group of genes with sequence homology and related overlapping functions. There should be homologous sequences and overlapping functions. The homology actually it means there is an evolutionary relationship. Sequence similarity and homology were different. Similar sequences were not always evolutionarily related. So if you have a common ancestry, common evolutionary history, then they are homologous. And there should be homology and overlapping functions in a multi-gene family. The superfamilies are defined as a group of proteins or genes of a common origin with non-overlapping functions. So the difference between superfamily and multi-gene family is that in the case of multi-gene family we have overlapping functions, whereas superfamilies, the functions will not be functions of the concerned proteins will not be overlapping. If a group of proteins or genes contain a domain of common origin, it is a superfamily. A protein or a gene may belong to 2 or more superfamilies because anyway there is non-overlapping functions. So based on some structural similarity and some relationships, there may be proteins which can be placed in various families, not on a single one. So this is an example of a multi-gene family. Here we have a common ancestry, and it is the different family members with overlapping functions and overlapping overlapping functions and a similar or common ancestry is there in case of a multi-gene family. So these clusters or these different genes in this family were placed in different clusters, and there will be an evolutionary history or evolutionary common ancestor in their evolution. Now, this is about the superfamilies. Here, the immunoglobulin superfamily is an example. There are T4, T8, then immunoglobulin at the surface, that is then T cell receptors, HLA antigens, then HLA class 2 antigen, class 1 antigens. All these things were different types of immunoglobulin family members, which will have some structural similarity, and of course there is a functionally it will be different. It will, each and every member of this superfamily will have a different function, and there will be an evolutionary relation also in this family. So these are all known as superfamilies, in which we have a common ancestry, but structurally they are structurally and functionally they will be different. So this is about the superfamilies. So these are all the different types of genetic elements with common ancestry or with a common evolutionary history in eukaryotic systems, which were evolved preserving their functions, or evolved to have related or functions in the biological system. So this is about pseudogenes, which is actually genes or duplicated genes with a loss of function, or different genes with structural and functional similarity close placed in the close proximity, like gene clusters, and the other related genes which can be classified or grouped together as gene families, then gene superfamilies, etc. Why we are actually assigning them into a family, that is also important, because if you have an unknown protein with unknown sequence, then you can place them by knowing the sequence of that particular protein or DNA. You can place them in the character family by sequence comparison. So you can place it in the character family. You can predict the properties, how it will work, or what will be the structure, what will be the function of this unknown protein. That will be decided, especially using bioinformatics tools and other things. There is function of the unknowns were predicted based on this one.